Hello, Frank. How are you doing?
Good. Hello, Tiago.
Hello, we are live. And yeah, it's a great pleasure and honor to me to introduce Frank. I'll try to spell it right. I learned from time that's Frank Kjeldsen, right,
Yes.
Frank?
Yeah, that's fine. Yes.
Oh, yeah. So Frank Kjeldsen. So Frank, he's a chemist. He graduated at SDU in Denmark, UNSE, and he did his PhD in Roman Zubarev's lab in Karolinska Institute. So you probably are familiar about the lab. His big proteomics lab too, and he used to work a lot in trying to understand the fundamental mechanisms of how peptides fragmentate in ion phase, right, Frank?
That's correct.
With electron induction, right?
Yeah.
I think you developed what was the name again?
Electron capture dissociation.
Electron cap ECD.
Yes.
Electron capture dissociation.
And its sibling is the electron transfer dissociation, which is more known today.
Exactly. So after quite a few years there, so he stayed in Sweden from 2001, 2006, right?
Yeah.
And then he got a really nice grant from Novo Nordisk, if I'm not mistaken.
From the Danish research.
Yes. Oh, okay. Sorry. Danish Institute Council. So it's a SDU grant in 2009, and then he became associate professor at the Department of Biochemistry and Molecular Biology at the University of Southern Denmark, which was then when I met him, because I went to Denmark in 2009, and end of 2009. First, I worked with Peter Hubstuff, but then Frank, from 2011 on, I worked in Frank's lab, in Frank's group. So it was like a really, really nice time that we had together. We used to have like a lot of traveling from Denmark to Germany, right?
Yeah.
To have meetings. It was like really fun. A lot of good talks. And well, then he got a really, really important grant as well. So Frank is really good in getting grants. That's definitely. I wish I had learned from him. So he got like the ERC, the European Research Council, as a consolidator award to consolidate your group. So then, of course, his group got bigger and bigger and bigger. He became a professor. It's a full professor, right, Frank?
Correct. Yeah, full professor. Yeah.
Now he's full professor at the BMB, the Department of Biochemistry and Molecular Biology, from Southern Denmark University. And yeah, so he's basically working. He's still working a little bit on trying to understand the fragmentation techniques. But he's doing a lot of work in nanoparticles. So trying to understand how the nanoparticles interact with each other. He's doing, and I know that because his background in chemistry, and he knows a lot that, so he's also like trying to develop new methods using what is called metal proteomics, and also trying to use these techniques to develop a new method to enrich for PTMs, things like that. So I'm not taking longer time. It's Frank's time. I just want to say you again that I'm really happy that you accepted this invitation. I'm really, really happy that you can share a bit more about your work. So it's your time now, Frank.
Yeah, thank you very much, Tiago. It's a pleasure to accept this invitation. I'm happy to tell what I know about use of proteomics in assessing the interaction between nanoparticles and biological systems such as cells and organs. So this is what the talk is about. But I think before we dig deeper into the proteomics part, it's nice to get a grip on what nanoparticles are and why they are used in our society. So nanoparticle technology is a fantastic area that has fascinated me for years now, because the fascination lies in the fact that the properties that you have in bulk material, when they are degraded down to nano size, which per definition is particles in the range of 1 to 100 nanometer, they change their physical and chemical properties. And there's basically an endless possibility to generate all sorts of nanoparticles of different material and composition and alloys and so forth. And over the years, researchers have demonstrated a huge range of application fields. So this is a technology that is come to stay, and I bet it will have transforming power to our society and how we live our lives. I just listed here a number of application fields, and there are many interesting ones. One is in diagnostics and treatment of diseases, because a number of diseases, like many of the diseases of the brain, is difficult to treat because of the blood-brain barrier that is difficult to pass for different drugs. But putting drugs into nanoparticles may be the way forward to treat these diseases. There are many other possibilities where you can use nanoparticle technology in the clinic. Also, the surface area of nanoparticles is, of course, combined larger than just the bulk material, and with new catalytic properties, they can be used, for instance, to waste management, but also many other possibilities are available. Silver nanoparticles in particular have shown to be antimicrobial, so they simply kill bacteria and are used in bandage and other products. But I would say already at this point, the use of them should be made in a way when it's necessary to apply this. Then hardness, you can make material with very strong tolerance to stress and so forth. The electron band gap, which describes or which is important to generate solar panels, can be modified with nanoparticles. Electrical properties, magnetic properties, and you can make new materials that are better than what we know. So this is just some of the applications, so you can understand that this is an important field. And just to give you an impression about the market value, I found this prediction that was made in 2016 on just nanotechnology within nanotechnology in the bio field, and this year it was predicted that it would range up to $42 billion. So it's a huge market too. So this is definitely important. On the other side, and it's often like that with new technologies, we have seen this in history, that development can go so fast that we also forget about safety. And this is where I think my group can contribute, and I think it's an important area because there has been a lot of concern actually from governmental organizations, especially here in the EU, which I know best. There's a lot of concern about unintentional exposure to these nanoparticles, because they are found in, as I said, in many different products, and they are released to the environment. And what happens then? And what does it mean when humans, animals, and plants are exposed to these particles? So to me, what I wrote down here, this is very important. I mean, I will fight for sustainable nanotechnology by applying proteomics and investigating how nanoparticles can impact human health. There's a lot of product, as mentioned, and I tried to look up if I could find references to that, and I found this one that although it stops at 15, you can see there's a steady increase in number of products containing nanoparticles. And I just got the recent number from 2020, and today there are more than 5,000 products that you can buy containing nanoparticles, at least claiming containing nanoparticles. These 5,000, this data comes from this database called the Nano database, which is very interesting, and I can advise anyone with interest in this field to visit it. You can make a lot of different distribution searches, and I did some, which is shown here. What I want to show here is really two things that I find notable. The first thing is actually something that I find quite disturbing, and that is, I would say this is 3/4, 3/4 of all the products. They are not well described actually in terms of what nanoparticles then contain. So they contain no information about size, distribution, shape, what compound it is, and so forth. So for consumers, this is difficult to navigate in. The other part is then where we have knowledge, and as you can see, the black pie here or piece is silver, and so it takes up quite some space in the nano product world. And I have also, for that reason, done some studies with silver nanoparticles. All right, so here we have a distribution of where silver nanoparticles are mainly used. As you can see, the majority of the products are within health and fitness, and garden and home appliances contains also food and beverages and so forth, even goods for children. So there's a wide distribution, but health and fitness takes up most of the distribution. I want to show you something, some of the products that you can find on the internet, and this is with silver nanoparticles. And silver, as I said, has been known for centuries to be antimicrobial, and especially in the nano size, they work very well. So you can find a lot of gels. You can even find solutions that you're supposed to drink every day. And I don't know if you can see this, but it says it supports your immune system. I will say and stress this by now, I would never, ever use any of these products because silver is really a toxic metal, and I don't believe there's any documentation that it will support your immune system. You can argue that if you have wounds that will not heal and you have tried everything, I mean, diabetic people have poor circulation of their blood, and this can prohibit wound healing. You can have bandages with silver material and also gel that can help in this, but then again, we're talking about situations where it really makes a difference. I don't think this is something you should just use from time to time. There's also a lot of fabrics with nanoparticles on the market, and the idea here is to remove bad odor. So there's a lot of athletic clothing, jogging outfits, and stuff like that. I can tell that in EU, these products are banned from 2020. You cannot sell textiles with nano silver. But there's many other products you can find. Bottles for children with nano silver, a frying pan, and I just checked this morning. You can, and this is probably relevant in these days, you can actually buy face masks with nano silver. Again, I would not do that, and I'm really in doubt about their documentation and approval from the health organizations in their countries. But apparently they are sold, and you can get them shipped. So you can see there's a lot of different products, and therefore we are exposed to these particles. It's quite common to actually spray refrigerators with thin silver film. Again, the argument is the same, to remove bad odor and prevent bacterial growth. A few years back, Samsung launched a washing machine claiming to release silver ions during the washing of the clothes. I have not seen how this works, but this is the claim. It had a few comments from different organizations advising consumers not to buy this product. But I'm not here to just downplay nanotechnology. All in all, as I said in the beginning, there are really a lot of potential good applications of nanotechnology. My point is just that we should use them in a wise manner. And now I want to show you some interesting things from the literature. This is a description of how one could use magnetic nanoparticles to cure tumor or to kill tumors in the body. So the idea is to inject a patient with magnetic nanoparticles that are functionalized in a way that they recognize the surface of a tumor cell. So this is a brilliant idea. And then after some time, you will use MRI scan to ensure that these nanoparticles are then taken up by the cancer cells, after which you will expose the patient to a strong alternating magnetic field. And because the particles are magnetic, they will shake a lot, which will induce hypothermia and basically boil and kill these tumor cells. So this is a very interesting, and I'm sure that treatments like this will be quite common someday. The researchers tried this on mice that had cancer, and as you can see, it really made a difference to the mice after treatment. Here's a very simple experiment suggested by other researchers. So, as said, silver nanoparticles kill bacteria, and here they have grown two different types. One is Staphylococcus aureus, and this is, you can see here in B, this is the control. You have bacterial growth. Silver nanoparticles kill these bacteria, and they made a variant of those that are magnetic. And so this is written in Danish, I apologize. But they also have the same effect. So the idea was then to have a different way in treating platelets in hospitals. There are patients that need transfusions of platelets, and the idea is not to transfer bacteria from one donor to a patient because they're already vulnerable. And today you shine this solution with UV light, but this damage also the proteins of the solution. So the suggestion here is to inject these magnetic nanoparticles, after which they kill the bacteria and you can retain them with a magnet. I don't know if this has come into practice, but this is another idea at least. So you can imagine that there's many routes of exposure. We can ingest them. We can inhale nanoparticles. They are so light, they can stay in the air too. They can traverse our skin, or we can become injected with them. And as they enter the body, they will have a routes of biodistribution and reach different organs. So it's quite complex to understand what happens actually to these nanoparticles after they enter the body, and what impact do they have to different organs. So this is the interest field of my group. And as you know, cells are not static or enclosed containers. They really interact with the environment, taking up nutrition, releasing waste products, communicating with other cells. So there's a lot of things going on in a cell. And what we know by now is that as a nanoparticle enter the bloodstream or any other biological fluid containing proteins and other biomolecules, a degradation will happen around a nanoparticle called, funny enough, corona. So this is termed a protein corona. So this is proteins from our body degrading a particle. And this will work as a stealth for the nanoparticle. So it's difficult for our body to recognize nanoparticles as foreign objects because they just have our own proteins and lipids around them. So we're interested in understanding how this corona is formed, how particles are taken up, and what impact they do. In fact, it's quite natural, as mentioned, for cells to take up particles of the nano size, because it has been known for many years that cells produce microvesicles and exosomes, and they are small units packed with DNA and proteins and other molecules, and a recipient cell can easily take these particles into them. So systems are already in place in our cells for engulfing particles in the nano range. So this is probably one of the problems with these nanoparticles. How to investigate the impact? There are a range of traditional typical methods. For one, imaging using different types of microscopy, and it gives you a good indication. As you can see, the green dots here are actually silver nanoparticles. These are data from a study by Thiago, the host now, and there are other techniques that we apply, a range of biochemical assays, cell viability, ROS, ATP content, and many other assays. And they are good in telling a lot about the toxicity. Is it toxic or not? But it doesn't say much about why or how. And this is where we think we can do a lot in the future. And we like omics technologies. We think they can answer much more precisely why and how nanoparticles interact with our cells. We apply proteomics because it can give us information about protein-protein networks and enriched biological pathways. So our working hypothesis is actually that if this sustainable nanotechnology is to be achieved, it requires comprehensive insight into this nano-bio interactions, and we will provide this with proteomics analysis. So this is where we stand today. As mentioned, it's good to have different methods to assess toxicity, and we apply all of them, but this talk will be primarily on mass spectrometry. We apply also quantitative proteomics, and just to remind you what the quantity of proteins can allow us to say. It's pretty general, but it's also applied to nano-bio studies. And I've shown here a map of my hometown to the left, Odense, and it's just to sort of make the resemblance. Every city has a power plant. It's the power plant of the cell is the mitochondria. It has a city wall that could be the membrane. The nucleus could be city hall, and the hospital could be proteins related to detoxification, production, or construction industry is the ribosomes, and there's many sort of funny resemblance. The point is here that if you know, let's say in a city, that the city hired twice as many workers to the power plant, you will deduce from that the city is lacking energy, and now that will be taken care of. The same is going for cells. So we're counting proteins involved in different functions, and in that way we can tell what what the cell is doing in order to cope with any perturbation, and that perturbation could be from a nano exposure study. So this is basically what we're doing. Is there a way to say something general about the response? We have done a number of studies by now, and there are many resemblances, but there are also unique observations, and I will say that it really depends on the cell type, the size of particles, dose, and exposure duration. But it's sure by now that nanoparticles can enter the cell. Some will stay also for some time in the membrane, but many of them will eventually be taken up by the cell. We know they interact and mingle or disturb the function of the mitochondria, which raises the amount of ROS. ROS is reactive oxygen species, can form radicals that can lead to apoptosis. It can also lead, it increases the oxidative stress, leading to protein oxidation that will either misfold and function wrong or be degraded. We have seen a number of proteins being upregulated to battle this oxidative stress, so antioxidant proteins. We have seen metabolic adaptation and transcription factor activation. So there's sort of a range of effects from these nanoparticles, and I'll give some examples of individual studies. The first I'll talk about is an idea we got, I think 2 years back, because most of the studies in the literature is about how organs or cells are behaving after being exposed to one type of a nanoparticle. We were thinking, as humans and other individuals, we are living in a world not exposed just by one type of nanoparticle. We're probably exposed to many different types. We're also exposed to other contaminants. So in this study, we wanted to understand whether exposure of one nanoparticle and another contaminant would have any synergy. And so one common contaminant that most humans are exposed to is cadmium. Cadmium comes from the dirt and is heavily released from cement production and mining, and is released and spread around the whole globe. So we are directly exposed to cadmium from the air, soil, and water, but certainly also from our food, where we get 90% of our exposure, unless we are smokers. Smokers have 10 times higher content of cadmium because tobacco plants have a preference for accumulating cadmium. So smokers really have a lot of cadmium in their body. It has a long half-life in our body, and it's toxic primarily to the liver and kidney. So in this study, we decided to study liver cells. So we made a simple experiment where we grew liver cells, HepG2. It's a cancer liver cell line. So we had a control experiment. We had one exposed just to silver nanoparticles, one to cadmium ions, and then the combination of them. So we wanted to study a synergy, and synergy exists if the effect is larger than the sum of the individual experiments. And what we saw here was a very strong indication, because the viability drop in silver nanoparticle was about 25%. It was about 10% for cadmium, but the combination resulted in something like 70% drop in viability, so a dramatic effect, which was quite disturbing. We wanted to investigate with proteomics what was going on, and we found several distinct pathways that were regulated, but primarily what caught our main interest was that the observation that basically all the enzymes of glycolysis was downregulated. including enzymes responsible for pentose phosphate pathway. All this was downregulated, which leads, which was also confirmed to lead to a immediate reduction in ATP generation. We saw then adaptation to that ATP depression by raising the burn of fatty acids and the activity of the TCA cycle and electron transport chain. So the cell tried to adapt to less production of ATP from glycolysis by raising the activity of other parts of the metabolism. But it can be quite devastating to for a liver not to be able to perform glycolysis. And I brought this picture just to remind you the function of the liver in terms of processing glucose. After a meal, we know the level of glucose is high, which promote excretion of insulin. And the liver plays a central role in metabolizing glucose. It performs glycolysis, which leads to generation of ATP. If there are plenty of energy, excess acetyl-CoA will be made to triacylglycerol, which is basically fat, and transported to adipose tissue. So it plays a major role in liver. But I should say also that glycolysis play a major role in all organs. So if this is the situation for liver, maybe other tissues are also hurt by cadmium and silver nanoparticles. It may become even worse between meals, because between meals we have to remember that the liver is the main responsible organ for stabilizing the blood glucose concentration to about 5 millimolar. And it does so by breaking down glucogen, which is basically a storage of glucose, but it only have a limited reservoir. If we don't eat in between that, it has to make a process called gluconeogenesis, which is the synthesis of glucose from other components like amino acids or glycerol. But the important part here is that in glyconeogenesis, it utilizes 7 out of the 10 enzymes of glycolysis because it's the reverse process. So if these proteins are inhibited or in less concentration, we have a problem in stabilizing this blood glucose concentration. Inhibiting or reducing the activity of the pentose phosphate pathway is also a problem. The pentose phosphate pathway does basically 2 things. It synthesizes the precursor necessary for nucleotide synthesis, but in this content, probably more important, it also makes NADPH, which is a reductive equivalent that is used in many biochemical reactions. One of them that are of interest in this context is the reduction of glutathione. And this is made by NADPH that reduces the dimer into free glutathione that then can be used to detoxify radical oxygen species. And these are produced primarily from the mitochondria, here hydrogen peroxide that can lead to radicals, but glutathione, which is probably the most important antioxidant, will make sure that this is transferred into water. But it cannot continue if we don't have enough NADPH. So this can be a potential problem also for the cells. So we were having data now showing that glycolysis is inhibited and probably also NADPH. And we were thinking this is quite disturbing, but maybe also interesting. We were working with HepG2 cells, which is a liver cancer cell. And we know from Warburg studies that what is characteristics of cancer cells are a high flux of glucose. It really burns a lot of glucose to make the components necessary for rapid proliferation and growth. So the consumption of glucose of these cells are much higher than normal cells, where there's a much more moderate consumption of glucose. So a long-lasting goal in cancer therapy has been actually to inhibit this high rate of glucose metabolism. So we're thinking it's interesting to understand if the same phenomenon then exists for normal cells as we have seen for cancer cells. So we made another study and we exchanged cadmium to cisplatin because cisplatin is probably one of the oldest chemotherapist drugs, and it's used also in liver cancer. So that makes it even more relevant, and we combined it with silver nanoparticles. So we wanted to understand again, is there any synergy, and will a normal cell line and a cancer cell line behave different? So here's the data of the biochemical assays. Here on the left, a viability test. So we made a control. We made one experiment as previous with silver alone. We made one with cisplatin, 2 different concentrations, and then the combination. And one needs to pay attention here because the light gray is the normal cell line. And as you can see, compared to cisplatin alone and the combination, we didn't see any drop at all in viability. But the situation is quite different when we have the combination therapy for the HepG2 cell. If you note, the viability drop with silver was, 25% and maybe 15% with cisplatin, and it drops to about 50% here. So a strong indication, again, of a synergetic effect, but only observed for the cancer cell line, which was quite interesting. In terms of ROS production, we did not see a raise either by combining the 2 drugs in the normal cell line, whereas it raised in the cancer cell line. All right. So we want to understand what is the explanation for that, and so we applied inductively coupled plasma mass spectrometry, which is a branch of mass spectrometry particularly sensitive to measuring elements in the PBB sensitivity. So it can measure very accurately the amount of metals and other components. So we took cells and wanted to know how much silver and platinum the cells contained. So here first, this is the cancer cell line. As you can see, in the combination therapy, we have a raise larger than expected in both treatments, whereas we did not see an increase in the content of silver. But for some reason, when we combine the 2, cells accumulate larger amount of the cisplatin in this cell line. And that could explain the higher degree of toxicity for this cell. For the normal cell line, THLE2, we did not see an increase in the uptake of cisplatin. So that is probably the main difference for the observations. Making proteomics, because we wanted to understand the reasoning behind the observations. We quantified about 6,500 proteins. And as you can see, we listed here all the regulated proteins, silver alone, cisplatin alone, and the combination. And the number of regulated proteins simply exploded in the combination exposure study with HepG2. It also raised in the THLE2 cell, although we did not see any effect in the viability test. There are actually signs that more proteins are deregulated, also in the combined. The normal cell line is completely insensitive to cellular nanoparticles, which was quite astonishing too. So there's a lot of interesting data here. We made a comparison to understand what pathways we are talking about between the two cell lines. So to the left we have HepG2, and then we have the normal cell line to the right. What they have in common is that, again, as we have seen previous studies, energy metabolism and stress response really has its high responses here. What we also see is that some of the deregulation or enriched pathways are coming only from one source, like up here we have a mitochondrial dysfunction comes primarily from cisplatin, where glycolysis deregulation is probably induced by cellular nanoparticles, because we don't see much deregulation or any significant deregulation with cisplatin alone. So this is quite interesting too. In the normal cell line, we see almost a similar pattern, but to us, it was very striking that the deregulation of glycolysis was not present in the normal cell line. So maybe we're seeing the same pattern here as we did in the previous study. So putting them side by side, all the deregulated proteins in the combination therapy, we have, as you can see, a number of pathways that are unique to the HepG2 that we don't find any deregulation in THLE2 cells, which also is in line with the more sort of robustness of the normal cell line. But zooming in on glycolysis and picking out all the proteins, this is the 10 proteins from glycolysis. What you see is that with blue, they are basically all downregulated in the HepG2 cell line, whereas only one of them is downregulated in THL2, and that is pyruvate kinase. So it really it shows the similar pattern as we have observed with the previous investigation. We also know that we have a lot of ROS. We saw an increase in ROS, so reactive oxygen species, and if there's not if cells are not scavenging these reactive oxygen species, it will eventually lead to apoptosis and other damaging effects of a cell. So we were looking at upstream regulators. So this is basically transcription factors that could explain some of this. And NERV2, as you see, which is a transcription factor that is upregulated or activated in THL2 cells, but not in HepG2. And this is a transcription factor that is known to activate genes, go to the nucleus, activate genes related to scavenging reactive oxygen species. If we look at the protein abundances, we see in the top here, so everything from here and up is HepG2, and we can see that the majority of a number of proteins is actually downregulated. And these are proteins that are involved in metabolism of glutathione, which is this important antioxidant. They are all reduced, which could explain why this cancer cell line is not dealing that well with the increase in reactive oxygen species. On contrary, we see a number of important proteins upregulated in the normal cell line: catalase, superoxide dismutase, NADPH, dehydrogenase, that are also proteins that is known to lower the oxidative stress of a cell. So we think that combined with other observations, this could explain why we have a difference between the two cells. Tiago, I need to know if I mean, I can see I'm running out of time, and I can stop here. I only have a few slides left, but
it's fine. It's fine. You can keep going.
Okay, I'll do my best to. You also took 5 minutes in the beginning, so
yeah, it's okay. Don't worry.
Okay, good.
As long as you need.
We are approaching the end, and the last slides will highlight a challenge that I see in the progress of applying proteomics in these nano-bio studies. The complexity exists because we have about 200 different cell types in our body, and we have seen already how different cells respond to nanoparticle exposure. And so in principle, we need to test all these cells. At the same time, on the other side, we have a huge amount of nanoparticles with different size, shape, coating, material. So this matrix is just tremendous. And I'm afraid that the production rate of nanoparticles in our society will overweigh our possibility to make safety assessment. And we cannot do that with the current pace. We can only do that if we make advancement and optimization to our workflow. We need to cut some corners and be more clever in our way to analyze this. So my previous postdoc, Anja Billing, and I wanted to challenge this problem by setting up a platform that could then analyze a larger amount of samples in a greater speed. We collaborated with in this project with Ulla Vogel from the National Research Center for Working Environment, and they were already in their having a study where they have intractable installation of mice with a number of nanoparticles that are iron and cobalt oxides, because they are exploited in nanomedicine. And so mice were having these particles in their lung, and after some time, the lung fluid was extracted, called BALF, and so it contains both the proteins of the lung as well as cells recruited to the lung. And they had 166 samples, and we wanted to finish all analysis in 4 days. So the strategy was as follows. We wanted to test first our reproducibility, because we were applying a new, to us, instrument, a new technology of chromatographic separation. Then we made a discovery phase where we took a subset of samples to investigate in depth what processes were going on, and from that also to generate library that we could use in our screening phase to quantify proteins and make a faster analysis. We teamed up with Evosep and Odense that produced this instrument called Evosep 1 that can run very short gradients without much dead time and without much carryover. So all our screenings were made with 24 minutes gradients, and those data was held against or combined and used in combination with our discovery phase. So I'll move fast here. So first, the evaluation of Evosep 1, we studied protein. We selected 6 proteins that span 6 orders of magnitude. And as you can see, the technical replicates, they don't deviate much. So this was very rewarding. And when we measured the cross correlation between the replicates, it had an average of average coefficient of 0.98, which was very good to us. And we ensured that our CVs was within what could be expected in clinical research, which has a common threshold of 20%. And we could have that with a subset of proteins that did not have any missing values. If we increase the number of missing values between measurements, of course, it goes up a little bit, but not too much, actually. I think working more with this instrumentation, we can bring this even further down. Okay, so the short highlights of the study. It has a lot of information, but we quantified about 1,700 proteins from the discovery phase, where about 700 of them were differentially expressed, and we used FOSSI-C clustering algorithm to place proteins in clusters that showed similar response to our iron oxide or cobalt oxide material. And we got these 6 clusters with different sort of behavior, and I cannot come into all the pathways that lies in those clusters because there's a lot of data set. I'll just highlight cluster 3 because it caught our interest, because it showed a unique immune response produced by neutrophils. It's a phenomenon called neutrophil extracellular traps, or NETosis, which is composed by 2 sets of molecules, DNA actually, and they are histones, together with a number of proteins that has that are proteinases, elastases that can break down pathogens, because NETosis is a phenomenon that exists from these neutrophils to combat bacterial infection. What we wanted to know is also, when we then had our screening phase where we're running very short gradients and many samples, will we be able to also verify such a phenomenon? And we tested that, and we could actually find many of the components, many of the markers for this this effect. So this was very cool. We had other differentially expressed proteins, and we placed them in this radar plot, showing different responses to metal iron response and wounding, to homeostasis and other GO terms. What we could highlight or see, because this experiment was done at 2 different days, so top is day 1 and then the bottom is day 3, that iron oxide induced a more acute response, because you can see they're dominating by the red shadows that is iron and iron with a little cobalt, where on day 3 it's more the blue colors, indicating that now it's the cobalt, more cobalt containing nanoparticles that are responsible for a more extensive response. Lastly, just finalize this neutrophil extracellular trap, because I find it really fascinating. These neutrophils can go to the point of infection and open themselves up, as shown here, and release its DNA and its histones as a web, like Spider-Man, and then it will entrap microbes and ensure that they are digested with these proteases, and in this way it can combat infection in the lung. We think, and I'm stressing this is our hypothesis, that when it comes then to nanoparticle, there may be a problem, and that is this NETosis will probably not be able to neutralize in the same way as a microbe the nanoparticle, because it's a different thing. And what is known in the literature for chronic exposure to other contaminants is that more neutrophils will be recruited, and they will open themselves and release their content to the lung, and they will keep on as long as this condition is going on. So if such a nanoparticle is sticking in the lung for a long time, we can foresee such a phenomenon. And from the literature, we know that this can lead to amplification of lung inflammation and chronic lung diseases. But as said, we don't have any data backing this up, but it's a potential risk, as I foresee. This is the last slide, and I just want to thank you for your attention and patience. I went a little over time. I have a lot of collaborators that have helped me with the studies, and very good students and postdocs that have done primarily all the work. And I want to thank also the funding agency, the EU, for supporting this work. Thank you all for your attention.
Thank you very much, Frank. Really nice talk. It's really nice to see that it's more information is being gathered related to nanoparticles. I know it's like it's a major problem in Europe, US as well. I think here in Brazil we don't. I'm not sure, but I'm not quite familiar that the industries are already like mixing this food.
Yeah.
One thing that draw my attention, I don't know if you remember, this the first paper that we published in 2014.
Yeah.
Drawn a lot of attention, and I learned by that time that there is like a kind of a secret community of the people that that really loves nanoparticles, especially silver nanoparticles. And there was this guy that was like criticizing our work in the net.
Yeah.
And I remember that one thing, it was actually quite clever, that he said that, well, because they state that the nanoparticles are really good in killing tumor cells. And you're shown here that indeed it seems to be more toxic for cancer cells.
Yeah.
And most of the time, and that's actually a problem in this kind of work that not us but science does, because it's easier to handle immortalized cells because they grow fast. It's kind of a more careless cell. Right?
Yeah.
And then my point is, and as far I was making like a small research here fast, this THLE2 cell that you use it as a control, it's still an immortalized cell, isn't it? Or do you know that?
No, I don't think it is it?
Okay, yeah, I don't know.
Yeah.
My point is that no, sorry, yeah, that's the one. Yeah, my point is, have you tried or done this kind of work that we have already done for cytotoxicity, using like a primary cells or non-immortalized cells to see if indeed these particles are toxic for us?
I have not tried it yet. It is working with primary cells is more tricky, as you point out. It's much easier to work with cancer cell lines, and I agree that some of the criticism we got in the review of the first papers were stating that drawing conclusions from dealing with cancer cell lines and then to how this would work in humans in vivo is stretching it too far, and I see the problem in that, and we are aware of it. We have, it will be of our priority list to move it also into working with primary cell lines. In fact, I have a project on the desk where we will then harvest cells or tissues from rodents that has been exposed to nanoparticles, because there's also something with the design of growing cells and exposing them. I think you remember that there's so many things that go on after you ingest or inhale a nanoparticle, because it will get coated with different proteins, and what is then the uptake rate and so forth for different tissues. So in reality, I will look very much forward to get the data when we start this project to expose rodents to these nanoparticles and then harvest material from, let's say, 15 different organs and make a analysis, because that will reflect also the biodistribution and a more in vivo like. But of course, the criticism could then be that this is rodents, this is not humans. But of course, we have to start somewhere. So I think it's just important that we don't stress the conclusions too far, and I think we are careful not to say that okay, this is toxic to humans, but more use it as there's something that points in the direction that it could be toxic, because we see this in our cell cultures.
Yes. Yeah. And I mean, another interesting application for the nanoparticles that you've shown is is this one to treat like a blood prior transfusion, right? With this magnetic nanoparticle. But then the point is, we will never be sure that the silver is not being released, right? Or the metal itself.
Yeah.
That's a problem, because we know that it can be released from the nanoparticles. From the studies that we've done.
It's although one would consider silver as a very inert element, because it is inert, right? But in the nano size, we have seen ourselves, but also it is documented in the literature, that there's actually quite a bit of dissolution. So release of silver ion. And this is also something we touched upon in our first publication with you. How much of the toxicity comes from the particle, I mean, the particle itself, and how much comes from the release of silver ions, because silver ions is very toxic too. So, but I think we documented that also in the first paper, and I think this is also why it has caught some interest.
It draws a lot of attentions. One last question from my side is actually two. Sorry.
Okay.
If you got a conclusion, why giving together the silver nanoparticle and then the cisplatin nanoparticle, why the uptake is increased? Did you see that, like the molecular mechanisms, like if you upregulate a transporter?
We looked at some of the there are certain proteins that are good at transporting metal ions. I mean, to keep homeostasis for, let's say, zinc and calcium and other metals, and we were looking at these metal transporters, if they could be sort of inhibited or in less concentration, and then they would not be able to sort of rescue the cell from the toxic impact of cadmium and cisplatin. As I recall, it was not quite clear if the regulation was significant enough to conclude anything from it, but we had these metal transporters in our consideration.
Yeah, great. I had a question here with Evosep, but Giuseppe has one I'll say for him. Just a curiosity, where did you do this ICP-MS analysis, or you have one home?
I don't have my own. I have a very good collaborator here at SDU who is basically a historical chemist. So he investigates bones and from old skeletons and paintings and whatnot, to sort of understand from where they come and what has been going on. And so he has such an instrument, because he needs to quantify elements in very precisely, and when there are very little of them. But it's very useful. It's very useful in this type of research.
Yeah, it is. It is. Yeah. Oh, it's good that you have one like in your neighborhood.
Yeah. It's very convenient.
A lot of people are like congratulating you, saying that like really nice talk. And Giuseppe Pisano, you remember him?
Yeah, sure.
Yeah, he's like congratulating you as well.
Okay, thank you, Giuseppe.
Evosep is a very useful platform. You show that the identification of 1,700 proteins using this platform. Do you think that the deepness of this analysis can be improved to detect less abundant proteins? Even though it's a problem to say that you had like 6 orders of magnitude, wasn't it? In that one?
Yeah, that's what we had
quite impressive.
Yeah. I mean, we don't get the same depth, of course. We went from, I mean, in our discovery phase, we made extensive pre-fractionation with Helix to get the depth of the proteome. And then in our screening phase, we could not allow that because of the time constraint, right? So this was just one shot of the BALF sample. And still, I mean, we went from 1,700 to 400 quantified proteins, which I found was quite good, because many of the things that many of the enriched pathways that we found in our discovery phase, we could actually refine also significantly with our EvoSep system. So this was pretty cool. I think we can do better, and this probably has to do with what type of instrumentation we can use. We tried just preliminary study with a timsTOF Pro, another type of mass spectrometer, and we actually went up to 600. I mean, for the same sample. So so there are ways, and I think it can become even better. But we have to cut some corners in this type of research if we're going to manage using proteomics in a way where we can analyze a lot of samples in a limited time.
Yeah.
But in this project that I envision, where we will take, let's say, 15 or 17 organs, we need maybe 5 or 10 samples from each organ, and then if you have this many males and this many females of the sample, all of a sudden you have 3,000 samples, right? And you cannot run 2-hour gradients of everything. You will never finish.
Yeah.
So our aim is actually 10 minutes. We will see if we can boil it down to 10 minutes.
Yeah.
In one
So 10 minutes, one run.
One sample.
Okay. And how long it took, this one?
This experiment was done in 4 days.
Okay.
With everything.
Okay.
I think that was pretty good too.
But I mean, you had like 1,700 proteins for BALF, which it's not like cell, right? It's this fluid. So
yes.
I think it's quite good number, I would say.
Yeah. We were.
I'm not aware about the literature, but I mean,
I think it was pretty good. We should remember, of course, that irritated lungs will be more permeable and will recruit cells, different cells, neutrophiles, but also other cells will migrate to the lung to help the combat. So the lung fluid will contain both the proteins of the lung, secreted proteins of the lung, but also some cells, but the cell count number may not be that high anyway. So I think 1,700 proteins is quite good too.
Yeah. Giuseppe adds one more comment, asking if you think that ion mobility could help in this regard.
Yeah, I mean, having another dimension to it, so that we don't just have chromatography. I think that would help, and I think that's actually why. I mean, the timsTOF Pro has ion mobility. And maybe that's the reason why we got 50% more actually protein.
Yeah. There is like Luis Santos, he's always joining us. Hi, Luis. He works for Bruker.
Okay.
I mean, I never had time to put my hands on it, but the data seems quite impressive with this timsPROT Pro.
Yeah.
It's like.
We are intrigued about this instrumentation. We don't have it yet, but we wanted to, because we had good data on our existing instrumentation, so we sent exactly the same sample to Brugger, and they used also an EvoSep in Bremen, in Germany. So everything was the same, so we could make a direct comparison. I think we had different projects that we evaluated, and the project that I have described here was particularly good with the timsTOF Pro. My colleagues here at BMB had other projects where data was comparable with what we have, or just slightly better. So I think it also depends. I think the timsTOF Pro is really good when you have short gradients and a high flux of eluting peptides.
Exactly.
Because it's very fast.
They are using a lot with EvoSep. Maybe like the clinical proteomics it's a good application for this form. I don't know if you can see in your screen. Magno is asking a question. Magno is a professor in the University of Rio, and he's saying that great talk. There are some recently published work that shows identification and quantification of up to 500 proteins using direct infusion without the HPLC, right?
Wow.
In DIA acquisition and in field acquisition. So, do you think this can be maybe the next step? Should be, right?
It sounds fantastic.
I think we've learned, right, Frank, that the trickiest part is the LC, right? That usually gets problem. So the downtime in the instrument most of the time is related to the HPLC or the UPLC.
Yeah.
And
we started to run more LC with higher flow because it's less sensitive than that. I haven't heard about direct infusion and this many quantified proteins, but the problem will be that you don't have any dimension of separation. So dynamic range can be an issue in terms of, I mean, you will quantify the most abundant ones. In plasma, for instance, there are 10 orders in magnitude from highest to lowest, right? And if you make a direct infusion, I guess you will quantify transferrin and albumin and other of the main components. If I remember correctly, it's something like the 20 most abundant proteins is 90% of the material in plasma. So I think it really depends also on the sample. Cells doesn't have the same dynamic range; it's less, but
maybe with ion mobility, the separation proof, but I don't know as well, like a dynamic range. And another problem that I can think, it's like quantitation, right? You would need to rely on chemical labeling because you don't have like you cannot do label free, right? You don't have the chromatography to make.
And if you use chemical isotopic labeling,
that's the one.
It's difficult if you isolate. If you use direct infusion, you will have everything in one. So if you isolate one species, you will co-isolate other peptides that are also labeled. And then when you fragment them and you read out your quantitation, then most likely you will have co-isolated peptides contributing to that, and it makes it a little complicated to assess the quantities. But I mean, sounds interesting at least.
I think it's one of the ways. I think that we are in a time that proteomics and mass spectrometry.
And we can also save the money for an LC.
Yeah, exactly. It's quite expensive too. But maybe it's another way that we can go, right? A lot of technologies are emerging and getting more robust, and maybe this is the way. We need to make it more not simple, but reliable and easy to be applied if you want to go to clinical proteomics.
Exactly.
we need to save time. That's for sure.
Yeah.
Yeah. Okay. So I think we're done.
Okay.
And again, to say thank you very much. It's always nice to see you again, and I'm really glad that you accepted the talk, to give a talk, and say hello to people in the PR group for me.
I will. Okay.
Okay, Frank. Thank you very much.
Yeah. Thank you. Bye bye.
Take care. Bye bye.